component of it. But let's first discuss the science and some of the main elements about Dream. So, crowdsourcing, I don't know whether you knew, the first time it was kind of coined as a term, it's kind of a new term, less than 10 years old, was in a Wired magazine article in which the author, I don't remember the name, was discussing this example of a company that created shirts, but created shirts that were stamped by images that were chosen by the crowds that eventually were going to buy it. So it was kind of almost, they were going to like it anyway, right? So this guy coined the term and immediately went viral. But like many things that resonate with us in terms of what it means, sometimes it's not so easy to define. So there is a book called Crowdsourcing that put out a definition that is a methodology that uses the voluntary help of large communities to solve problems posed by an organization. So the elements here is an organization, large communities of voluntary workers, and a methodology. And so we will talk a little bit about the methodology here. But first, the fact that I love this quotation by Johann Bernoulli. So 300 years ago, more or less, he said, oh, I have to be here for the rest. 300 years ago, he said he wanted to screw up Newton because he was a disciple of Leibniz, and they had invented the calculus, right? And so they wanted to put a problem that Newton couldn't solve with his fluxions and so on. So he said, he put out the debate out, which is the problem of the brachistochrone, as a challenge for the mathematicians of the time. And he said, we are all assured that there is scarcely anything more calculated to rouse noble minds to attempt work conducive to the increase of knowledge than the setting of problems at once difficult and useful, by the solving of which they may attain to personal fame, as it were by a special, unique way, and raise for themselves enduring monuments with posterity. For this reason, I propose to the most eminent analysts of this age some problem by means of which, as though by a touchstone, they might test their own methods, apply their powers, and share with me anything they discover, in order that each might thereupon receive his due meed of credit when I publicly announced the fact. It looks like a dream challenge almost, but that was in the late 1600s, 1700. There is a famous prize that was the longitude prize, in order to know where you are east-west at sea. In the 1800s, chemical engineering got a very seminal piece of work by Nicolas Leblanc that resulted in processing water from seawater, soda from seawater, and in the 1900s, the aviations. Now, more recently, we have seen other community efforts. One is the X Prize, another is Innocentive. I don't know whether you guys know. These are companies that profit from crowdsourcing, as Kaggle does. Wikipedia, some people say, is not a crowdsourcing really. It's a community organizing itself to write articles and so on. But in any case, it's kind of what the online communities can enable. More in the area of computational biology, I think we all know and respect the work that CASP did in the area of structure prediction, and that was particularly inspiring to me, that in 2005 and 6 we started to think of how to validate network inference, and we created DREAM, that stands for Dialogue for Reverse Engineering Assessment and Methods. And We have also been involved in Improver, which is a version of Dream, but more targeted to industrial community. So Dream is purely academic and non-for-profit. Actually, the Dream Challenges is a community that is completely non-centralized, and little by little, people from different parts are starting to uh, converge to do challenges, as we are going to tell today. But really, there is no links in terms of institutional glue. But that is good and bad. And so, more recently, we joined forces with Sage Bionetworks that gives us a structure that it will be transparent when we talk about Synapse, what we have accomplished together with Sage when we organize Dream challenges. So crowdsourcing, you get a problem, you try to target the community, they propose solutions, and in principle you solve the problem. But in Dream, we are more talking to some extent about benchmarking, in which you have a generic problem, like for example, find who responds, who doesn't to some particular medicine in a particular disease, and in one instance you know the solution. So you crowdsource and you get proposed solutions, and in that instance you can know which is the best solution. And in order to figure out who are the most likely methods to solve that, and then hopefully those same methods will generalize to cases that for which you don't have the solution. So this is also very useful for people because they can know how well or how not so well their algorithms work. I always like to remind the audience when I give this talk to know, Dream is not just a competition to declare a winner. There is more than that. It's clearly a method assessment that everybody can take advantage of. Of course, we try to discover the best methods, but also we are trying to discover the solvability of a scientific problem. Sometimes we have data, we have a problem, we try to solve it. If I cannot solve it, it doesn't mean that it's not solvable. But if none of 100 teams can solve it, then it means that there is something missing in it. I mean, it's not proof that it's unsolvable with that data set, but it means that there is something that probably is ill-posed. Either data is not enough for that, or the questions are ill-posed. We sample the space of methods because of these 50 teams or 100 people or 200 people that participate. Different people are enamored with different kinds of methods, and then we can see how those methods in their hands work to solve these problems. We like to think that we help in terms of acceleration of research, in that people working on these challenges, 100 people at a time, would do in maybe in 4 months what hardly any lab could do in 10 years, in testing how to analyze this data for a particular problem and so on. So really, it's a lot of people trying to solve a problem, and if we should learn something from it, positive results and both negative results as well. And lastly, we are very much about building communities around problems. We are trying to make sure that people that have similar interests get together, talk to each other, reinforce their skills and methods by talking, because usually, even though sometimes it's not exactly like this, we compete with each other rather than collaborate. We are mostly trying to make sure that our competitors don't publish earlier than us. We have to try to, if I produce data, try to keep the data to myself and not share with others, so that I can milk that data before anybody else. So that probably there are other ways to work in which data is democratized, everybody tries to look at it, and maybe we can help patients when there is a translational problem. So it's a different mindset, right? And I think that both have to exist, but to some extent these days I think we are much more in the field of trying to compete with each other rather to collaborate. And Dream is trying to create a framework where collaboration is the currency. And finally, if you put together all our solutions in challenges, we have seen very often that the ensemble of the solutions works very well. So we'll discuss a little bit more about that later. So in order to to make sure that we get those goals, we created the Dream challenges. And I said before, in February of 2013, we put together the community that we're working on Dream with the structure that SAGE provides through their Synapse platform, but also through their similar vision that science should be open and data should be open and so on. And so we got together, and I think that we have synergized very well. We have produced a lot of challenges, and well, we'll see more about that later. So, as I said before, the mission of Dream is to contribute to the solution of important biomedical problems, and the problems that we have solved or addressed so far are here in the bottom part of the slide. Transcriptional signaling networks is a key part of what we do. I think that most of the people who have participated in Dream are particularly interested in this area, as I am particularly as well. But also we have done other areas and are very interested in extending to how to model the response to a perturbation, how to parameterize kinetic models, how to infer parameters that Julio and Pablo have worked a lot on, and also translational research such as toxicology, response to rheumatoid arthritis, medication, Alzheimer's disease, ALS, AML, breast cancer, and so on and so forth. So those are the areas. So we try to contribute to the solution of these biomedical problems. We try to foster collaboration between research groups, democratize data, accelerate research, and objectively assess algorithm performance. And this is the growth of the participation in Dream in terms of submitting teams. We started with about 50 in 2007 in the first Dream 2 challenge, and by 2014, when we did the Dream 9 challenges, we got to about 350 participants. The reason why we call Dream 2 is because it was the second conference, actually. There was a first conference, that was Dream 1, but there was no challenge. The challenge started at Dream 2. So all the challenges, and I think that this is a little outdated, there are 35 challenges ongoing now, are available through the Synapse website and through the Dream website itself as well. So you can go there, get all the data, get all the algorithms that were used for scoring, and look at the description of the challenge and the literature around it. We will talk about this a little bit later, but basically there is an art and a science about organizing these challenges that over the years we started to streamline a little bit better. So some of these things go parallel, but here is more or less the idea. First, we have to work with a community of domain experts to understand what the scientific questions are for a particular domain, be it cancer, be it toxicology, be it response to drug perturbations. Then we assemble a team of people that are interested in that particular problem, that have the skills, and we get the data, curate the data, run a dry run in order to see whether the problem is solvable and to what degree, create the web infrastructure for which Synapse software engineers work and have done a terrific job in creating an infrastructure for these challenges. We think of incentives. How do we do in order for people to participate, right? Why would people participate? People ask me this, when you give talks probably as well. Why would people participate in a challenge, right? And so, well, then we'll see later why. We want to know.
That's why the Guinness is really important.
Guinness is very important. There is also pizza. No, we are going to make pizza available for you guys. It's a cheap proposition, right? Because we are free. We need to think about advertising and communications, how to, and then we open the challenge after a lot of work. Some people at some point told me, well, organizing challenges is very easy, right? You just get data, put it out, and that's it. Actually, there is more to organizing a challenge, and that's so. But when we open, we are all very excited about, how the forum is nucleating the questions and people dialoguing with each other. Eventually, the open phase closes. We score, which there is a lot of method to the scoring. And then it's time to analyze the results of the challenge, the submissions, which methods worked, which methods didn't. And sometimes from the methods themselves and the results, we understand that there are, for example, genes that are better predicted in terms of response to perturbations, or compounds that are better predicted in terms of their activities, and so on. Then we write a paper, we put the challenge results in a database, and move on. And that's one of the things that we'll discuss later, is what we call the long tail of the challenge. What happens after the challenge, how people can use it, and so on. We have been rather successful in publishing in high-profile journals the results of our challenges. Last year alone, these are the covers: Dream 5, Dream 6, Dream 7. We didn't get any other cover, though we tried. But for example, last year we got, I think, 3 papers in Nature Biotechnology, one on synergy prediction, one on drug sensitivity, one on ALS. This year already we have a Nature Methods. What else? Pluscom Bio. Nature Biotech Impress. So we have been really successful. And there is something to be discussed later about what we have called challenge-assisted peer review, which is a proposition that editors have grown to like, at least in the Nature community. Okay, let me talk a little bit about network inference, because I want to discuss, this was kind of the origin of why we started to do that. And really we want to think, while we are very open and embrace machine learning, I think there is also a community, and I am one of that community, that think that mechanistic understanding is very important as well, not just machine learning solutions, but a little bit more mechanistic. Even though sometimes this mechanistic understanding produces results that are worse than machine learning, but and therefore we still insist, and we will see it later in the challenge that Julio will be presenting on networks. What do we use networks for? Probably I don't have to tell you, but integration of data, enrichment analysis, causal inference, master regulators, upstream reasons why differential there are genes that are differentially expressed, and so on. So usually you get data from a variety of places, but typically we have used data from gene expression or proteomics. We did some challenges with flow cytometry, but not with ChIP-seq. We hope we will. Mass cytoph is one challenge that we are organizing, or at least we hope that we will get to organize. All kinds of perturbations are going to be important, and in one of the papers that we wrote, we saw the importance of having as wide a landscape of perturbations as possible. Eventually, we infer the network from this, and we don't have any idea whether the network is correct or not, and so that's why we want to benchmark in one way or another. So who knows here Arachne, which is one of the most better known network inference algorithms? You guys don't know it? Never heard of it? So let me tell you about what Arachne does. The reason why I want to present this it is one of it's one of the most used, not necessarily the best. Actually, it didn't score the best in our network inference, but there is something simple to it, and and it has been validated biologically several times. So I wanted to discuss it a little bit because it's cute. So basically, it's trying to find the mutual information between pairs of genes, and typically we try to identify which are the transcription factors, transcriptional regulators, and which are the targets. That could be transcriptional regulators or not. And then here the thickness of the arrow represents how high a mutual information is. Who knows what mutual information is? Everybody knows, right? Yes. So here is the formula, and basically what it is how much information in one gene there is with respect to the other, but it's symmetric, so we cannot in principle use mutual information for directed network inference, but there are other ways to overcome that with using prior knowledge. So the other thing that basically Arachne does is to try to get rid of some of the false positives, because everything will have some mutual information with something else. So the idea is, how do we determine what is the arrows that we have to get rid of? First of all, a null model is created that says if you have a mutual information less than a threshold, then probably you are not an edge. So let's get rid of those, and that's where we get rid of those. And then we try to see which arrows are explained by other arrows. For example, if you look at, by the way, what we use there is what is called the data processing inequality, which is a theorem that works when you have, for example, Markov chain that has information that goes from one state to the next and to the next. And so if you not one state, but one node to the next and to the next. So if you have a mutual information between, or a connection, a communication between 1 and 2, and a communication between 2 and 3, for example, edges in a network, then the mutual information between 1 and 3 is smaller than the minimum between 2 and 3 and 1 and 2. So if you use that might suggest that maybe here, 1 and 3 are not connected. So if I have that this edge, we don't have a pointer, right? You have one? Okay. If we have one here that is smaller mutual information than 1 and 3 and 3 and 4, then we might want to flag that for to get rid of it. But before getting rid of it, we have to see whether we also can explain some other pieces of the network. But here, for example, this guy dominates over these two. So we may need to so this is bigger than these other two. So we may need to keep it. But if you see that this one also can explain this one, then we don't need that one. So we get rid of that one. And now this guy can be explained by this and this. So and again, this one can be explained by this and this. And what you end up is with a sparser network that might have some chance at being correct because it's based on some assumption this data that the data processing inequality is correct. It's not in general. So this may fail. But this is what Arachne is. And we published this in 2005 in Nature Genetics. In the same year, I published another similar network inference method that was also very nice with some colleagues at IBM. Published, with Andrea Califano, this one, and with my IBM colleagues, the other ones in bioinformatics. And because this was published in Nature Genetics, it became viral, and so it became very, very prevalent. So Andrea has been using this to a beautiful amount of other methods that could explain a lot of the things that he sees and make predictions that eventually he validated. But one thing that we have to recognize is when we wrote this paper and we created a lot of edges connected to the transcription factor MIC, we supposedly validated it, but the way in which we validated it was took the 12 most top predictions, and the ghost came. Ghost. No. We took the 12 highest mutual information predictions, and we checked those, and that's a very easy way of checking for a validation. You really have to take a big chunk of a data set and validate at random there and see what is the rate of success at a given sensitivity. So I wasn't really happy with the way we validated this. At that time, there were lots of, we started this in 2005, and when we started to look at network inference, there was kind of an exponential growth. And I didn't look at 2010 to 2015, but there are lots of papers that address the issue of the problem of network inference. And so, but there were so many, we really decided to do challenges that could determine which methods were the better. And we went to in 2008, 9, and 10, we created three network inference challenges. In total, there were 91 teams that made 806 network predictions. So I want to discuss a little bit this Dream 5 network inference challenge that we published in Nature Biotechnology, Nature Methods, in which we talk about the wisdom of crowds, how aggregation of networks does well. So in that case, what we had is from different labs, mostly Jim Collins' lab, we had an Equality data in which we had 805 arrays and 487 conditions, and we had 296 transcription factors and 4,200 genes. We had also data for yeast, we had data for Staphylococcus aureus, and we had an in silico challenge, as we had done in Dream 3 and Dream 4. And I'm not presenting today one data set that we did also on using phosphoproteomics data, but this is more transcriptional regulation. Now we gave data for this. There were 160 arrays and 53 conditions for S aureus, but we didn't have a gold standard to check it. But it was an interesting exercise because we wanted to show that the wisdom of crowds could be used to infer it. So anyway, we gave all these conditions to anybody who cared to solve it, and there were 29 methods that were applied to it, and there were 6 off-the-shelf methods, including Arachne, and we tested them all, and we got networks for each of them, and let's see how they did. So we wanted to see whether there were methods that were doing better than others, and actually that was a very revealing exercise because we realized something that we kept seeing in other dream challenges, and probably we should have known this, but there is no best method really. The best method in the in silico was different, was a regression method, was different from the best method in E. coli that was a statistical method based on an ANOVA comparison, was different than the yeast network, which was more based on random forests. And if you aggregate everything, if you aggregate who did best across them all in some way, you see that the best was neither of the best in the individual ones. Was the method that was the most consistent across all the subnetworks. But most importantly, the best scoring network was the one that was created out of aggregating the community networks of each of them. So, for example, in silico, the community network, which was aggregated in a way that I will make a little clearer later, um, was the top scoring. It wasn't the top scoring in the E. coli network, and it wasn't the top scoring in the yeast network, but it was amongst it was the third best scoring and was the fourth best scoring in the yeast. Overall, it was the best scoring if you aggregate all of them. So there is something about the aggregation that renders the aggregate network better than or at least at the top. And usually you don't know which is the best. Therefore, you would not know which one to use. So it's a good strategy to take the aggregate. I love these plots because it shows the concept of ensemble predictions and wisdom of crowds so clearly. If you look at the solutions, this is the overall score. This is minus the log 10 p-value of the aggregate network. And if you see, the best performing had a 40 as the overall score, the worst performing had 0. When you aggregate 5 at random amongst these 29 networks, you get that the median is much better than the median of the individual. And if you aggregate 10 at random amongst these, you get even a better scoring. And if you aggregate 15, the median score is better than the best of the individual ones. And finally, if you aggregate all of them, you get the best possible prediction that you could with the networks that you had here. So there is something else that I think is also relevant here, is that how often, if you aggregate, would your network be better than the best? And this plot answers that question. So let's take, for example, aggregating 10 networks. And let's see, the fraction is this one. How often, if you aggregate 10 networks, the aggregate is going to be better than the 10 networks that you aggregate? And the answer here is 60%. But if you allow it to be the best or the second best, is so in 85% of the cases, and in 91% of the cases is either the best, second best, or third best. If you aggregate 20 of them, in 80% of the cases, the aggregate was better than the best. And if you allow for it to be better than the best or second best, it's so in 99% of the cases. So, guys, we have to aggregate, and that's a way to say, let's work together, with different people that have different methods, and eventually understand why one method and the other aggregated perform better than the individual ones. There were some interesting validation of the aggregate network for E. coli. So remember that in the E. coli, we had that the best network was the aggregate network was the third best. So we tried to aggregate. So we went there and we made a cutoff at 50% precision because we knew the gold standard. We had a cutoff using the Regulon DB edges, and we found that cutoff gave us 50% precision. So that contained 1688 edges, and so we went to take 1, 2, 3, 4, 5 transcription factors and stimulated E. coli, giving what you need to give in order to stimulate these transcription factors. For example, here L-rhamnose, or in this case copper. And so there was the following experiment done at Jim Collins' lab. You stimulate the network and see whether the putative targets of these transcription factors go up with respect to another E. coli for which this transcription factor had been knocked down, or knockout actually. So we had an experimental way to see, and what we observed is that the new edges that were predicted in the in the community network were validated at the 40% amongst those 1688 edges. So you go basically take 5 transcription factors, which are all over the place, take the edges that are daughters of these transcription factors according to the community network, and you go and validate experimentally. And we got 40%, which is pretty much what we expected because we knew that we took those numbers from. So I think this is the right way to validate. You don't have to validate the top of your predictions. That's too easy. That's good. I mean, you are validating something, but you are not validating your method. To validate your method, you have to say, we have error bars. The error bar is the precision, and then if you predict that this is the precision, then you have to go and try to validate and see whether you get the same precision. In the case of the staff Oreos, we didn't have really, this is wrong numbers, but we didn't have really the gold standard, but we looked at the new edges coming from transcription factors that were known to be related to pathogenicity. And basically what we are saying here is, even though we didn't validate those, there is some good chance that these new edges connect unknown genes to pathogenic related processes. Okay, so I think we were supposed to finish this at 9:30, right?
Yeah.
So I want to go very fast through another challenge that we did in 2012, which is called the Nci Dream Challenge. There will be a second challenge that I will discuss in a highlight talk on Monday. But right now I only want to say what we did in this Nci Dream about predict drug sensitivity in cell lines. And this was done using Joe Gray's breast cancer cell line data. And this was a big group of people. Jim Costello did the lion's share of this work. Misha Menden also, probably he should be closer to the top. This is one problem with these dream challenges. Everybody has a claim to credit, right? But we have too many people. So we are discussing with publishers how to do in order to assign the credits in a way that is not unfair to people who work very hard but actually could not be on top, in the first ones. Anyway, I got people very angry here because they were not close to the top. But this is the thing as it is. We work very closely with Dan Gallahan, Dina Singer, who are in the U.S. The director and deputy director of the cancer biology program, and they are loving the concept of dream. So this challenge was basically consisting of the following. We took 53 breast cancer cell lines, and there were a lot of very interesting baseline data. We had copy number variations, RPPA, phosphoproteomics data, methylation data, exome-seq, gene expression data, and RNA-seq of the cells before any perturbation. We had the GI50. Who knows what the GI50 of a drug is? So just in case, I'm asking not to see who doesn't, but rather, should I explain it?
Yes.
Okay. The GI50 is the concentration of a drug at which the growth of the cells are 50% of the maximal possible. So the drug lowers how fast these cells grow, and when you find the drug response, the concentration at which the growth is inhibited by 50% is called GI50. So we had a GI50 for all these 53 cell lines, and we gave, for the 31 drugs that we had, a training set of 35 cell lines, and we kept 18, and we asked people, can you predict for each of these drugs which is the cell that gets the most, that is the most sensitive, and the least sensitive in an order list, and then we will check whether your order list coincides with the measured list. But we had to take into account here the fact that measurements are noisy, right? So you have an error bar, which was not small. So we had to, in this case, we wanted to use something similar to the concordance index. Sorry, something similar to the Spearman correlation, which is the correlation of the ranks. But because one of the ranks were noisy, we decided to use something similar to the Spearman, which is called the concordance index, and we modified it to have into account the errors into something that we call the probabilistic concordance index, that had into account that by chance, even though the average for this particular drug, this cell line is in average more than this, the error bars are such that really, if you predict the opposite, it has a chance to be true as well. Of course, this one with respect to this is very disjoint, so this should be more sensitive than this one. Anyway, so this is the probabilistic concordance index under the null hypothesis will have this distribution, and these are the results that we obtain. So you see that the results are not all that good, right? This is a very important problem in precision medicine. Given a biopsy, can we predict what is the best drug? Or given a drug, can we predict the best cancer for which this drug repurposing? But if the average concordance index in a random prediction would be 0.5, the best prediction here had an FDR of 10 to the minus 5. It's not bad, but it's not all that good. The best possible prediction, considering the errors, would be here. So we have this gap. So this is what we call, the problem of the half full or half empty glass. So we are doing much better than random. We are picking signal in the data, but so the glass is half full. But with respect to how well we could be, the glass is half empty. So we should, but I think this is encouraging that we should continue to work on these kinds of problems. We can see here that the data shows that even though the best performer was a kernel method based on multitask learning, did pretty good actually. The second one was based on nonlinear regression, and was a completely different method. It was a random forest-based method. But you see that the third one is here. Is another type of method that I right now don't remember. So again, there is no one method that dominates over the others, except maybe this that didn't have any method. So actually, nonlinearity seems to have been an important factor in the modeling of this, and we discussed this in the paper. We also tried other methods for scoring. We did we threshold the sensitivity and say sensitive and non-sensitive, and the best performer was about 78% balanced accuracy in sensitivity, which is not bad, right? But you can't see here something that we will see many times in dream challenges, that according to the weighted probabilistic concordance index metric, the best performer is this guy, but this guy is second best performer with respect to balance accuracy. And we will see that something that during scoring we should discuss the fact that we need to use more than one metric. We'll not linger too much on this, but this was the idea of the best performing method. He used different views. He took the data and looked at it in all possible ways, binarized, ternarized, took windows based on genes that are part of pathways that are handpicked and so on. He combined the views in a combinatorial way. he discretized the views, and so all that he gave to under different kernels, using multiple kernel learning, and learn the weights in the training set, combine the kernels, and eventually got predictions for the GI50, and then use that method to test set, and he did pretty well. Here we can see that if you aggregate the best predictions, as we said before, again we see that the best goes for a probabilistic concordance index that is worse than the average random up to this, which is the best performer. But if you aggregate the 5 at random or 10 at random, you again see the same as before, that the median goes up. And if you aggregate in order, and there is a problem with doing it this way because this is a supervised way of doing it. But suppose that we know by a prior that these are the best in order. So if you aggregate like this, you are going to get, even though the individual methods are doing worse and worse and worse, we still get some boost in our ensemble prediction. And even if we aggregate the whole of them, we don't do much worse than the best of all. So again, and the rule of thumb for us has been 7 to 10. You get, if you aggregate 7 to 10, that you know that are reasonably good methods, you get the boost in your prediction with respect to the best. Okay, I'm not going to discuss this challenge because we will present it. There are a couple of challenges that are here that will be presented on Monday, if you're interested in it. One is predicting drug synergy. The other one is predicting response in ALS patients. I just want to say that we have run these challenges last year on Alzheimer's disease, AML, outcome prediction, and rheumatoid arthritis, and these two as well. And we are still, we have just finished running this one that Pablo organized, the Dream Olfaction Prediction Challenge, that went very, very well in several respects. I think that we had, for the first time, really being able to predict how a person is going to smell an odor based on the odor chemical composition. But most interestingly, the problem again is half empty or half full. People got together and said we have to work together and try to understand why some methods work better than others, and I want to use your method in order to improve mine. So they are almost self-organizing in the forum, and probably we will have a collaborative phase. We have an ongoing prostate cancer challenge going on. We haven't finished that. We are opening the final submissions for final scoring. This is based purely on clinical data, and the question is, how long will prostate cancer patients going to survive? One important concept about this is based on clinical trial data. It's hard to get pharmaceutical companies to share clinical trial data. They would rather share their toothbrush, I think, than share their clinical trial data. But our colleagues at a foundation called Project DataSphere were able to pull together data from different, what is called the comparator arm of clinical trials, and using that we created a data set together with Sage and Project DataSphere and Dream that created the data set for the prostate cancer Dream Challenge. And we are still trying to figure out how to call somatic mutations from BCF files. I think this is an interesting one. We are working on ALS2 stratification challenge that is just been announced. Here is my half empty, half full. Depending on which fish you are, you will say that the glass is half empty or half full, right? Some people are really pessimistic, you know, and some people are very optimistic. I'm very optimistic, so I see always that there is signal in the data. We should keep going. Lots of people working on this. I don't want to mention them all. Sage BioNetworks has played a fundamental role. My colleagues at IBM Pablo is here, have played also a very important role. Julio's team at the EBI have played a fundamental role. You know, Brian, you are not here. We should put you here.
I was on the first slide.
You're in the first slide. This is one problem. You know, this great that there are so many people. I always make mistakes and not putting important people. So people from University of Colorado, Oregon Health and Sciences, Prize for Life, NCI, Columbia, and so on and so forth. So this is really collaborative, and